organizer of International Conference on Probiotics and Prebiotics by Magnus conferences. I am Dr. Manisha Mandal, Assistant Professor in the Department of Physiology, MGM Medical College, India. At the outset, I would like to express my gratitude for giving me the opportunity to present my paper before you, the title of which is Metagenomic Analysis of Non-Dairy Fermented Foods Giving Food Source Associated Structure-Function Differences in Taxonomy, Biosynthesis of Secondary Metabolites and Xenobiotic Degradation. Recent studies have suggested that healthy diet should include fermented foods to enhance probiotics as life beneficial microorganisms in our gut. The need for non-milk based fermented foods has arisen in lactose intolerance, those allergic to milk proteins, in vegans due to high fat and cholesterol content in dairy products. However, the mechanism underlying the health benefits accruing from the non-milk fermented food microbiome are not clearly understood. The current topic deals with the microorganism of fermented food, which mostly contain beneficial bacteria including probiotics. Therefore, it is important to explain the mechanism of action of probiotics leading to health beneficial activity. This figure explains different mechanism of action of probiotics. Firstly, it modulates the immune system by enhancing antibody response, by reducing inflammation, and by stimulation of phagocytosis. In addition, it interacts with gut microbiota through production of antimicrobials by cross-feeding and substrate transformation, as well as it supports microbiota stability. Besides, probiotics produce organic acids such as lactate, propionate, and acetate, thus reducing colonic pH. It also enhances nutrient production through cross-feeding. It also plays role in colonization resistance by competing for nutrients and location. In addition, it improves barrier function by stimulating mucin production and supporting epithelial cell health. Probiotics also interacts with the host mediated by cell surface structures such as pymine, LPX, TG binding proteins, mucin binding protein, toll-like receptor ligands, lipotecoic acid, and exopolysaccharides. It also manufactures small molecules with systemic effects such as neurochemicals including cortisol, serotonin, gamma amino, butyric acid. It is also involved with the synthesis of tryptophan and histamine derivatives as well as satiety hormones. It also produces conjugated linoleic acid. In addition, it produces enzymes like bile salt hydrolase and lactases. Thus, these are the diverse mechanisms which are likely to drive probiotic benefits to host health. So with this objective, the present study was carried out to attain a comprehensive insight into the structural and functional composition of microbiomes by metagenomic analysis of a diverse range of non-dairy based fermented products traditionally used across a number of countries. To achieve this, the methods adopted were V6 single and fast-2 sequences of 16S ribosomal RNA bacterial genes were first using ISRA toolkits from NCBI containing 23 non-dairy fermented food samples from 9 datasets, including 3 sets of pickle samples, 3 sauerkraut, 2 whole wheat, 3 sets of spontaneously fermented sourdough and fermented sourdough by inoculation, 2 samples of Arabian foods, 2 samples of olive, and 3 samples of fermented carrot juice, and 2 pozole samples. The first 2 sequences were then imported to Kind2 in Miniconda 3 and subjected to demultiplexing quality control with DADA2 algorithm phylogenetic analysis, and taxonomic classification using Green Genes database. Statistical analysis for community profiling was achieved with alpha diversity, beta diversity, and core microbiome analysis. Clustering analysis was carried out with heat map to compare abundances of different taxonomic levels, dendrogram correlation analysis, and pattern search. Differential abundance analysis was carried out with univariate statistics, and marker gene survey was done using metagenome sequence. Finally, PCRUST was applied for prediction of functional potential and pathway analysis of the microbiota from 16S output. All these above calculations were done in Ubuntu 20.04.2 LTS 64-bit operating system 3368 genome version. Now the results. The study revealed significant association between taxa abundance with fermentation source as well as food types. So fermentation source and food types were the major drivers of microbial composition. And this figure shows the alpha diversity with pozole and whole wheat demonstrating respectively the highest and lowest effect on microbial diversity. While inoculated sourdough exhibited highest species richness. This is the plot of beta diversity, which shows the similarity of parameters based on 95% of all samples drawn from underlying Gaussian distribution of microbial communities at taxon level. A single large confidence ellipse was obtained under fermented liquid food encompassing other food types. Besides, two other confidence ellipses belonging to pickles and sourdough types of food were also obtained. Functionally, pickles and dough exhibited more diversity and richness respectively, while sauerkraut displayed no effect on microbial community functioning. Illumina sequencing of 16S ribosomal RNA amplicons yielded approximately 23.4 lakhs sequences with mean of 1.02 lakhs reads ranging from 18,000 to 2.83 lakhs across the 23 samples that were beamed into 532 operational taxonomic units. A total of 6 phyla were detected across 12 families, which included Proteobacteria 21%, Firmicutes 18%, Cyanobacteria 10%, Actinobacteria 2%, and less than 1% each of Bacteroidetes and Tuleriquetes. The majority of classes were Bacilli, Alpha Proteobacteria, Gamma Proteobacteria, and Actinobacteria. This is the taxonomy at genus level. Lactobacillus was consistently abundant in carrot juice, spontaneous sourdough, and in pozole, while Lactococcus, Leuconostoc, and Basilia were abundant in carrot juice only. These are the 4 beneficial lactic acid bacteria with established probiotic potentiality. This slide shows the coordination of the bacteria at genus level. So, the top 5 correlated bacterial genus included Marina microbium with Vibrio, Bradyrhizobium with Polynucleobacter, Pseudomonas with Clostridium, and Pelomonas, Caestobacter with Rhodophyrax. Their correlation coefficients ranging from 0.85 to 0.93. PCRUST analysis showed dominance of KIG metabolism, including that of amino acid, carbohydrate, energy, cofactors and vitamins, nucleotide, terpenoids, polyketoids, besides biosynthesis of secondary metabolites and xenobiotic biodegradation, as well as their metabolism. Genes related to ATP binding acid type transport system were dominant, essential for nutrient uptake, cell viability, protein secretion, signal transduction, drug and antibiotic resistance. The presence of genes related to degradation of atrazine, styrene, geraniol, limonene, pinene, toluene, which might be useful for the restoration of environmental quality were found. The study also revealed the abundance of genes related to the biosynthesis of penicillin cephalosporin, flavonoid, and enzymycin with possible industrial application. Mapping of KEGG orthology showed enrichment of 246 pathways, out of which top 10 pathways have been demonstrated here. The corresponding KO names have been indicated on the left, while the values in bracket indicate the involved number of KEGG orthologs. The top 10 pathways include related to that of metabolism, microbial metabolism in diverse environment, biosynthesis of secondary metabolites, bacterial secretion system, biosynthesis of cofactors, benzoate degradation, protein export to component system, porphyrin metabolism, and degradation of aromatic compounds. KO mapping showed significant expression of modules, for example, ubiquinol biosynthesis from cordyceps to ubiquinol, which is under cofactor and vitamin metabolism. Besides, ivermectin biosynthesis was also significantly expressed under the category of macrolide biosynthesis belonging to terpenoids and polyketides. In addition, aerobactin biosynthesis from lysine was also significantly expressed. This slide depicts some of the KEGG orthologs, that is 15 in number, which were expressed in the degradation of aromatic compound pathway. These are highlighted in red. In addition, 19 KEGG orthologs were significantly expressed during benzoate degradation. Besides, 12 KEGG orthologs were involved in amino benzoate degradation. Now, the conclusion. The most abundant phylum, class, order, family, genus, and species were respectively Firmicutes 64%, Bacilli 58%, Lactobacillus 55%, Lactobacillus 37%, Lactobacillus 10%, and Z 9%. Ozol and whole wheat possessed highest and lowest microbial diversity respectively, while inoculated sourdough exhibited highest species richness. Functionally, pickles and dough were more diverse and rich respectively, while sauerkraut displayed no effect on microbial community function. Lactobacillus was consistently abundant in carrot juice, spontaneous sourdough, ozol, and whole wheat, while Lactococcus, Leuconostoc, and Basilia were abundant in carrot juice only. The fourth, beneficial lactic acid bacteria with established probiotic potentiality. Bacterial strains of the genus Pseudomonas with projected bioremediation capacity was dominant in spontaneously fermented sourdough. On the other hand, involvement of pathogenic bacteriogenida such as Clostridium, Corynebacterium, Pseudomonas, Escherichia, Rhodococcus, Streptococcus were found in pickles, which cannot be ruled out for major health concern to the consuming population. The genes related to the biosynthesis of secondary metabolites and xenobiotic degradation were in abundance. So, non-dairy genomics may be applied for monitoring the microbial composition and functionality to obtain candidate non-dairy starter strains towards development of functional food with desirable gene traits and assessing the safety of the traditional non-milk fermented foods. So that was all for my presentation. Thank you for your hearing and thank you for your patience in listening. Thank you.